Materials needed: U-2 OS cells in a culture flask or dish, sterile PBS without Ca2+, Mg2+, trypsin EDTA 0.05% or 0.25%, complete DMEM, Dolbeco's modified Eagle medium, plus 10% FBS plus 1%, pen Strep, sterile conical tubes, 15 or 50 ml, new culture flasks, dishes, pipettes, plus sterile tips, cell counter, optional, biosafety cabinet, CO2 incubator, centrifuge, optional. Step 1. Wear gloves and wash hands with 70% ethanol. Then check confluency. Observe cells under a microscope. When U-2 OS cells reached our 80–90% confluency, they're ready to split. Overconfluent cells grow slower and may differentiate. Step 2. Put complete media, PBS, Trypsinization into hot water incubator for 30 minutes. Wash hand, eye, biosafety cabinet, and all. The required materials such as pipettes, PBS, TEC media with 70% ethanol. Four, remove old medium. In the biosafety cabinet, aspirate the spent medium completely. Three, wash with PBS twice. Add 5 ml PBS for T25 flask, scale up for larger. Gently tilt to wash away serum residues, which inhibit trypsin. Aspirate PBS completely. 4. Add trypsin. Add 1–2 ml of trypsin for T25, enough to cover the cell layer. Gently rock to distribute. 6. Incubate. Place flask in 37 degrees C incubator for 2 to 5 minutes. Check under the microscope. Cells should round up and detach. Tap the side of the flask gently to help them detach. Take 4 culture plates, then label it properly. Make new cell culture plates arrangement before by transferring 8 ml complete media to each plate. Step 7. Neutralize trypsin. Add 3-5 ml of complete DMEM to stop trypsinization. Pipette up and down several times to make a single-cell suspension. Decide split ratio, common for U-2 OS, 1.3 to 1.6 every 2 to 3 days. 9. Seed into new flasks. Example, if 1.4 split, add 2 ml of cell suspension to 8 ml of fresh medium, 10 ml total for T75. Gently rock the flask to evenly distribute cells. 10. Incubate. Place flasks in a 37 degrees Celsius, 5% CO2 incubator. Check next day. Cells should attach and spread. Clean the biosafety cabinet with 70% ethanol and stay safe and happy. Thank you.